I can't picture, like, you know, the character Hiccup. I just can't picture
him like as
Jay
a
Baruchel
leader.
but Jay Baruchel with a beard.
Oh my god, he can't fucking grow. I bet he has like 3 pubes. Like he has no hair on his body.
Okay, as someone who's like...
That's what's wrong with his voice. He's stuck in puberty. Welcome to the Bumble Bears podcast. It's Willie and Saz here.
In what might be our last recording in our OG home.
In our OG home, where we are backyard beekeepers who have a keen interest in all things bee related. And yes, we are in the process of moving. That's part of the reason why this is belated.
There's many reasons why it's belated. Because...
The first one is that we're disorganized people. Second one is that I am super pregnant and I don't feel good very often. And what else, Willie? We've been busy, haven't we?
Yeah, we have had a monumentous birthday week and baby shower.
Yeah! We're now 32 and we still don't have our shit together. But yeah, this is probably the last time we record in this house because we are moving and there's more carpet and wallpaper in our new house.
Ooh.
So it's probably going to be a little more of a hushed acoustic, which is actually good for this kind of thing.
You're the sound expert.
Well, and we're not going to be so close to a busy road as we are here, which I don't think the mic picks it up. Speaking of mic picking things up, upon listening to our previous recordings, I have realized, and this is probably reflective of true life, you come across very strong and clear, and then I sound like I'm like whispering. And that's, I don't like talking loud. It stresses me out, okay? I tend to speak quietly because I got anxiety, bitch, okay? I don't really like raising my voice, but I'm going to try to be louder now.
And I can't control the volume of my voice.
That's true, yeah. That pretty much sums us up. But anyways, Willie, what are we talking about today?
Cool. So this is the second time we're going to bring the science edge to it, to the point where I think it is now worthy of its own segment, and also segment mini introduction, which I would just aptly call Bees in Science. And this is... We touched on this briefly in the past, if for our loyal listenership of 5 people.
Of my sister.
Yeah, and...
Hi, Becky. Love you.
And I think Samara also
Hi,
listens
Samara.
Yeah, hey, Samara. And we don't know who the other 3 people are, but to our very loyal listener base of 5 people, you would have heard us, I guess, give a bit of a review and a tribute, because this is a tribute, you gotta believe it, to people which, mix science with bees. And so we spoke about the discovery of, the giant Wallace bee last time and how cool that was. And we also... the startup company in Nigeria, how they had this new futuristic, modular hives. So
these are a heavily researched creature, aren't they? Because they're so interesting, because they have such a complex social structure, and because they're so important to, agricultural and the survival of living things. They've been quite heavily researched, I would say.
Yeah, so we're going to dive back into it as well. And just as Sarah was saying, and to add on that, what makes bees awesome is they are the 3rd insect to have their genome mapped behind the Drosophila, aka the fruit fly, and the mosquito. So the fact that we have mapped the genome of the honeybee means that if you were to use them for biochemical experiments, you can use, like, say, basically um, gene manipulation techniques, or understand, like, the genetic machinery as to why biological processes work. And that's very important in science, which I won't go into right now, but trust me on this.
Okay, so we have two recent and very interesting papers that have come out regarding bees. The first one, Willie, do you want to introduce the title and authors?
Yes, I would love to introduce those papers. So, this is a paper in the Journal of Lab Animal, which is part of the Nature Publishing Group. And the title is called Honeybees as Models for Gut Microbiota Research. The lead author is Hao Zheng, and the corresponding author is Nancy Moran. Nancy hails from the University of Texas, so massive shout out to Nancy. She has been slaying science for a very long time. She's a professor of integrated biology. She got her PhD in 1982. So basically, she is.
What? She looks so young. Unless that headshot's from like 20 years ago.
No, you know what? I think it's because she's surrounded by animals and plants.
Yeah.
So she lives like
she
a good.
She looks stoked and healthy and.
Yeah. And, she has, a never-ending list of accolades and awards. I envy her science-ness. If I could be half the researcher Nancy could be, then I would have done well with my academic career. So, massive shout out to Nancy for driving this paper, which we're going to touch on. So, Sarah, what did you like about this study?
Actually, this one I find, I mean, they're both really interesting, but this one speaks to me more because, as we have discussed several times, just throughout our life together, both being interested in nutrition and the human body and stuff, it is a very important and growing area of research to understand gut bacteria and how it affects your entire livelihood, like in so many different ways. And basically, so this paper is about gut bacteria in honeybees and how they've been able to research it in depth and how they have discovered how it affects, many different aspects of the bee's livelihood. And then how that can also be reflected in humans. One thing that's coming to mind right now, we have a very dear friend who unfortunately suffers from lupus, pretty hardcore, and there's kind of new research saying that maybe the gut bacteria is affecting the lupus, or maybe it, has some kind of correlation to how the lupus affects you and stuff like that. And our friend has said, like, that kind of stuff is actually, he feels like that might be true as somebody who, is living with this disease. So if, the more that we can understand this kind of stuff, hopefully people who have autoimmune disorders or other, like, really difficult issues to live with, maybe we can help them.
Yeah, because our dear friend did actually say that when on the most recent trip, abroad in Japan, they said, unlike even eating fast food in Japan, it's like symptoms not as bad.
Yeah.
But back home in Melbourne, it's it's touch and go. Yeah. So there is
because he has a different diet here than when he goes to Japan, and then he's eating lots of fish and seaweed and fresh stuff like
and
that.
Even what they define
it makes a big difference on his symptoms.
Yeah. So even what they define as junk, as like a post midnight bowl of ramen, he goes, yeah, it's just it doesn't cause your symptoms to flare up as badly.
Which is interesting because, as we know, there are quite a difference in occurrences of certain diseases, like in Asia and Japan and stuff like that. And a lot of people say, like, it's because of their diet. So, maybe there could be some answers there. Also, before we moved in together last year, I went and saw this doctor in the States. I've had a rough time with doctors. They don't listen to you. They just want money. We don't have Medicare, so you have to pay everything.
But you have Medicare now.
Oh, yes. I do. It's quite joyful. But, anyways, I found this doctor who does a little bit of, I guess, like, alternative and different therapies and stuff like that. And I just thought something's not right. I felt like I'm not digesting food as well as I should. I'm tired, blah, blah, blah. And when I described everything to her, she goes, maybe your gut bacteria balance is off, and why don't you try this diet? And it basically was like a low glycemic index diet, high protein, high fat, and then she said take these fluconazole. What was that?
It's like that's antifungal, right?
Yeah, yes, it was an antifungal. And then, so she said 8 weeks, do this. It's going to suck at first, but then you're going to feel like $1 million afterwards. And you know what? That's exactly what happened. The first 2 weeks, I thought I was going to die.
I remember those messages.
Yeah, I felt lightheaded, I felt sick, and
you had no energy at first as well.
Mm-hmm. And she kept saying, it's because the bad bacteria is dying, you just have to, like, keep with it, stick with it. And then after about 2 weeks, I woke up one morning, and I was like, I feel reborn. I feel good. And the rest of the time, I felt amazing. Like, it changed my life. I kind of wish I had stuck with the diet, to be honest, because I totally fell off the bandwagon after. But, I mean, that's the kind of thing, it's starting to become mainstream, but before it really wasn't.
Yeah, because we always, there's an age of, like, saying you are what you eat. And, like, there is a lot of, the more we research it, there isn't a lot of truth to that. And in essence, what they think lies the key is, you've got this giant, you have your intestines, which is like a massive amount of surface area, which...
There's just colonies and colonies of little...
Yeah, and it's not just a mechanical operation of just breaking down food and absorbing nutrients, but it's a very, very high emerging, fast-growing field. They're learning more and more about it, because especially with over 7 billion people in the world, we're not all going to digest food the same. Like, I have the guts of a raccoon, as you've probably noticed, except I do have my kryptonites, which cause me to absolutely slay the toilet.
Lentils.
Oh, man. And also that weird tofu thing, which...
That was so sad.
Yeah,
Really
which, that was Montezuma's revenge on, like, that was the worst. But the listeners, our 5 listeners, don't want to listen to my diarrhea stories. So, but in essence, your guts basically aren't just there to absorb nutrients and push out chocolate snakes. They actually play a large part in your immune system and even your mood and, like,
how well you sleep.
Yeah. And even, there's a researcher, Monash, she actually works on how the microbiome actually might, determine the risk factors of you getting an infection in your brain following a brain injury.
Mm-hmm.
So...
It's all connected.
Yeah, so I won't go into it for this podcast, but if you ever want to read up on Connie Wong's research, she's from the Hudson, formerly known as the Monash Medical Center. She does a lot of awesome work on basically how the gut microbiome can infiltrate the brain when the blood-brain barrier has been compromised.
And then on that same, like, kind of, I guess, rant about this whole thing, the doctor that I saw, one of the things she asked me when she was kind of assessing me, she said, did you take a lot of antibiotics as a kid? And I did. I was sick a lot as a child. And she was saying that it is suggested that, when you're a child, obviously you're still developing and stuff, and that if you are taking tons of antibiotics, that could end up killing off a lot of like your gut flora and messing you up in adulthood. So very interesting. But anyways, Willie, why don't you get into the specifics of this article?
Cool. So what the and if Nancy's listening, I hope I'm doing your paper justice. But basically what they're suggesting as to why the honeybee would make an excellent model for studying the microbiome is firstly, they've been, I mentioned before, the genome's been sequenced. So all the transcription pathways, all the signaling, which is required to say go from process A to process B, that can be mapped. We can try and identify what genes are responsible for that. So we know what genetic machinery is possible for any sort of process, if we can observe them. Bees, especially during spring, the queen can lay something like 2,000 to 4,000 eggs a day. So there is no shortage of sample size, say, versus like if you were to go use a mammalian model, like say a mouse.
What's their gestation is like a few weeks, a month.
Yeah, like you still have the rough 3-week gestation, from like embryonic period, but a mouse is only pushing out like 8 pups at most, right? Like you've got 2,000 emerging workers. It means you can absolutely high throughput any sort of experiment of interest. One thing which also simplifies things, and I'll backtrack a bit to explain. In the general science community, you have 2 methods of trying to address a question. You can go bottom up, so ground up, which means starting from simpler mechanisms and then going to understand the basics, then going into more complicated systems. And this is what this research is doing. Or you can go top down, and that's so starting from the clinic. An example would be say Parkinson's, and then trying to backtrack and figure out what's going on. So with research like this, we're working from bottom up, and bees don't have as varied diet as mammalian systems. So the fact that their diet basically just contains of pollen and nectar.
I don't know. I saw the Bee Movie, and he drank coffee and ate cake crumbs.
And Cinnabon?
Yeah, the Cinnabon.
Yeah, well, there is nothing factual in the Bee Movie.
Except that Jerry Seinfeld's not funny.
Yeah.
But anyways.
So the fact that bees just consume nectar and pollen, albeit from different, flora sources, but they're essentially eating like 2 types of foods, then you don't need as diverse of flora to exist in the gut to break it down. Therefore, with less combinations and permutations, we can try and understand the signaling pathways, with better detail.
Yeah. And it's like starting with a simpler, I guess, like how you start with simpler mathematical sequences and then you build on them, right? It's kind of like that. It's let's look at it at the absolute simplest base.
Yeah, like, with like when you're a child, you get introduced to Lego Duplo, which only has like giant blocks. And then there's Lego Basic, then I think Lego System, and I think Lego Technic is, I think, the highest one.
Damn, well, you know the whole Lego system, don't you?
Even though I was never any good at building Legos. So because starting with a simpler system, we can try and understand we can try and fully understand the chemical and genetic machinery responsible for the microbiota. Then we can move up to a more advanced system. Other major advantage of the honeybee as a model is that when they're in the pupa stage, because they haven't eaten honey yet, their gut starts off microbiota free. So
yeah, which you wouldn't be able to model that in a human, because even Lammy in my belly right now has already developed some digestive system. So like
it's infiltrating from the yolk sac.
Yeah.
And Lammy is absolutely guzzling down the amniotic fluid, and that first shit is going to be disgusting.
Well, you better hope so, because you're gonna change it.
Yeah. So in that regards, like, you can literally start from like time point zero. And the other thing about, say using honeybees, as a potential model for to study the microbiome is, in theory, you could just do it, in one hive because you are building out so many workers. Like, you don't need to start like, you don't need to set up like, hundreds of, mating pairs and stuff like that. This one colony would be enough to sustain your research.
Yeah.
So I do hope that this does absolutely take off. I do read the paper. I know it's really heavy in a lot of zoological terminology, but I do think it's very well written that the general audience can digest most of it. You might need maybe dictionary.com to translate a couple of words, but I think most of you won't.
Because didn't they also say that they were able to isolate some of the bacterial strains and like duplicate them in a controlled environment so they could actually study, the life cycle of the bacteria itself?
Yeah, absolutely, yeah. Like they could, they can culture all that stuff. They can do so many things, just from basically isolating the cultures within the bee guts from like immature and mature bees. So I think it's an excellent model. I would happily work in this.
We are not
field.
moving to Austin.
No, but maybe...
Actually, Austin's supposed to be like the only cool part of Texas, but still, it's Texas.
So moving on, our next paper, our next is titled "Honey as a Biomonitor for a Changing World." Lead author Kate Smith with, I believe, Kathy Gordon as the corresponding author. And this was published very recently in Nature Sustainability. And Sarah, what did you find neat about this study?
What I find neat about this is that we are constantly discovering new facets of what bees can do and how they can help us understand the environment and just what a complex and fascinating creature they are, because these researchers were able to basically analyze honey from 6 different locations, and they found a scale measure that reflected the amount of lead that was in the honey and how it reflected the amount of lead that was in the pollution in the air around the environment where the honey came from.
Yeah, and in the soil as well.
Yeah.
So they, what's great about this, and this is also another emerging way that people are doing science, and this is absolutely fascinating to me, is this potentially can be like citizen science done on a global scale. And for those who don't know what citizen science is that a lot of questions requires a lot of sampling, and even a big lab at most only has 20 odd people. So you can't possibly send 20 people to scour everywhere. And Everywhere. So what we want to, some projects like this, what would be awesome is, and there's lots of intelligent people, out there, especially say in Melbourne. We live in a city of roughly 4 million people. And a lot of people, are just happy to contribute, to the good of mankind. And what this, Australia actually has some really good citizen science initiatives. If you just Google citizen science Australia, there's actually a lot of projects which any one of you can actually partake in with no science training required prior.
Like they'll take you out in the field and be like, okay, this is what you need to do.
Yeah.
Here's the little coring device or whatever, and like.
Yeah, like, and even not that, like, that's maybe like, getting on the really extreme end of things, but some things could be like, hey, are you planning like a holiday to the Otways?
Bring us some rocks.
Yeah, they'll be like, tell us which path you walk in, and if you see any koalas, tell us where you've seen them, and we'll put them in the map so we can try and figure out their...
see koalas.
Yeah, and then they can, map this sort of stuff. So that's like, mobilizing, that's what they call citizen science. A hilarious project, which is available, in Melbourne, which I don't know if they're going to get too many volunteers, but they want to look at how antibiotic resistance is. Their suggestion that antibiotic resistance might be spreading, jumping species, and not just restricted to humans. And on one hand they think it's because of veterinary sciences, because a lot of livestock and cats and dogs get antibiotics as well. But they think it's trying to get it spreading for other means, and they don't know why. So they are calling for volunteers to collect possum poo from around the neighborhood.
Do you know who would love to do that? Tanyan.
Our little man.
He'll be like, "Can I roll in it and bring you the poop swatch on my collar?"
Yeah, so they need to mobilize people and collect possum poo, put it in a bag, so that, I think they actually have a kit for you to do the experiments at home. Or you can just send it to the lab.
Why not? Let's do it. When Lemmy's big enough to, walk around and talk and stuff, and be like, "All right, Lemmy, we're going to go possum poo hunting today."
Yeah. So I love the concept of citizen science. I know we're a bit elbow deep in other things right now, but I implore any of you that, you don't have to it doesn't have to be like, they do have some extreme projects. They do say, like, if you're going to Mount Kosciuszko, we're worried that, the soil concentration is going to shit. So please take us a sample from the highest point of Mount Kosciuszko. And obviously that's hard. But then there's, some great ones where, say Roeville Lakes. It's just like, oh, we want to know if the bacteria in the soil, in the lake is changing. Please get us a sample from
We
that.
want to know how many tinnies of tuies are just floating around in the water.
Exactly. So, as this study was suggesting, they used they had beehives in 6 different locations. They looked at the honey, and it was a very good scale representation of the relative lead concentration in that area. So, hypothetically, if you were to upscale the amount of beehives you take honey samples from, you could have a very, very high resolution biomarker
concentration map kind of thing.
Yeah, fingerprint. And then, it allows you to do preventative things of like, oh no, like between year 0 and year 4, tin concentration went up by X%. Why is it doing that? And
because of tinny fish.
Because of tinny fish. So, do you have a read if you
Because they only did this in like a small scale, obviously, because it's like, the first kind of study of its kind. But they just did this in like a small area of British Columbia.
Yeah, they did it in 6 areas in British Columbia in Canada. There's a lot of things which are very heavy in science in it, which is basically what you have to for nature sustainability. But using the most advanced mass spectrometry methods available, they were able to reproducibly show that the honey, from a hive is truly representative of, any sort of contaminants which are in the soil. So a lot of the heavy science is mainly to validate the way they went about analyzing the honey. But the take-home message is basically bees have been around well before us. I hope they'll still be around well after us, because,
they deserve it.
They've
done nothing but good for this world.
Yeah.
And what have we done? Nickelback.
Well, that's also from Canada.
True.
How the hell do we wind up like this? Why weren't we able to stop polluting the seas? This can be spread, to basically every major city, and I think it's very neat.
It is very neat, because lead's bad. I mean, obviously, metals and heavy metals have their place in this world, but when it comes to ingesting lead, that is not a good thing. So if we can, it's just so cool that we can use bees to...
They're our scouts.
Yeah, to measure the concentration of pollutants. Like, it's just fascinating. Yeah, it's like having these little living army of homies.
Yeah, well, they're basically very in tune with nature. That
It makes me wonder what else, what else are we gonna figure out that bees can do?
Yeah, like the honey that was sampled in this study. Well, okay, I know we said lead. Lead is very, very toxic, but it's in very low concentrations. Like if it was in, say, if it was from right outside a petrol refinery, that honey would probably kill the hive. So we're talking fractions of nanograms here, but still, as you're all aware, once you ingest lead, you can't actually get it out of your bloodstream. So it's a good kind of a preemptive warning on how we can still rescue our environment before it is literally gone beyond repair.
Yeah, I don't know. I think it might already be headed in that direction because the polar ice caps are melting. Trump is the president of the U.S. It's bad, okay?
It is pretty bad. But thank you so much to these guys for basically going full science and going extreme chemistry to see like another very useful tool for honey in the grand scheme of science and trying to improve the world. Well, that concludes Bees in Science for this week. And yeah, if you see any other sort of bee related study
send it to us.
Yeah, which you think we should read, definitely send it to us. And how could you get in contact with us? Because I don't manage that side of things.
You can find us on Instagram at BumbleBear Farms, or you can email us at bumblebearfarms@gmail.com. Yeah, it's really cool because both of these papers came out this year, right, in 2019.
Oh, this one came out very recently, March 2019. The other one was, I think the first electronic publication was October 2018, but in print it's only come out this month. So, yeah, it just makes me excited to think that two really groundbreaking and really fascinating articles have come out in the bee world this recently, and obviously I'm sure these labs are still thinking, okay, how can we build on this research? We're going to probably do a follow-up, hopefully, see where they go with this, and I just want to see, what's going to happen in the bee world next. Yeah. Until next time, yeah, keep sciencing, and I hope you guys, especially here in Aussie land in the Southern Hemisphere, coming into winter, I hope you have lots of minimal lead honey, to put on your delicious porridge. Yeah, in your life. And we will next cast from our new house, which has nano wallpaper, and you won't hear that fucking creaky door that's been going off this whole time that drives us crazy. I don't have a sign out, so I would just say, farewell, and enjoy living life with the bees. This is how you remind me of what I really am. Imagine being Nickelback. Imagine Jay Baruchel singing Nickelback. Okay, bye everybody. Bye. Look at this graph. Every time I do it makes me laugh. We measured all the lead. And what the hell is on Joey's head?